Good morning, everyone, and welcome to today's discussion. The question we're exploring is a profound one. Is animal testing necessary for medical advancements? It's a topic that evokes strong opinions and touches upon complex scientific and ethical considerations. We are privileged to have three distinguished experts with us today to share their perspectives. Dr. Evelyn Hayes, a medical researcher, who will argue for the continued necessity of animal testing under strict regulations. Mr. Samuel Green, a bioethicist, who will focus on the ethical objections to animal testing. And Dr. Lena Petrova, a scientist, who will discuss the advancements and potential of alternative testing methods. Our format will begin with opening statements from each of our guests. This will be followed by a moderated discussion and rebuttal section, and will conclude with closing statements. Let us begin this important conversation.
Thank you, Alex. I want to start by acknowledging that the ethical concerns surrounding animal testing are valid and deeply felt by many, including those in the scientific community. However, I must assert that at our current stage of scientific understanding and technological capability, animal testing remains a necessary component for achieving significant medical advancements. Historically, the path to many of our most critical medical breakthroughs is paved with research involving animal models. Consider the development of vaccines for devastating diseases like polio and measles, or the discovery of insulin, which transformed type 1 diabetes from a fatal condition to a manageable one. Organ transplantation techniques, which have saved countless lives, were refined through animal studies. These are not trivial accomplishments. They represent monumental leaps in human health. The scientific community operates under a stringent ethical framework, commonly known as the 3 rupees: replacement, reduction, and refinement. Replacement encourages the use of non-animal methods wherever possible. Reduction aims to minimize the number of animals used in experiments. Refinement focuses on modifying procedures to minimize any potential pain, suffering, or distress. Furthermore, strict regulatory oversight governs all animal research, ensuring that studies are scientifically justified and ethically conducted. The fundamental challenge is that the complexity of a living biological system, how a drug interacts with different organs, how the immune system responds, or how a disease progresses systemically, cannot yet be fully replicated by available alternative methods. While in vitro tests and computer models are valuable tools, they often fall short in predicting these complex whole-body interactions. Our primary goal as medical researchers is to advance human health and alleviate suffering. Currently, responsible animal testing plays an indispensable role in achieving this vital objective, especially for ensuring the safety and efficacy of new treatments before they reach human patients.
Thank you, Alex. While Dr. Hayes highlights the perceived necessities and historical context, I believe we must fundamentally challenge the moral justification of inflicting suffering on sentient beings for human benefit. The core of my argument rests on ethical principles. Animals are not mere tools or resources for human use. They are sentient individuals, capable of experiencing pain, fear, and distress, much like we are. This brings us to concepts such as animal rights, the idea that animals have an inherent value and a right to be treated with respect, independent of their utility to humans. We must also confront the issue of speciesism, which is a prejudice or bias in favor of the interests of members of one's own species and against those of members of other species. Is it ethically consistent to subject non-human animals to procedures we would never consider performing on human beings, simply because they belong to a different species? The greater good argument, often invoked to justify animal testing, becomes deeply problematic when the burden of suffering is placed upon non-consenting subjects. These animals do not volunteer. They do not understand the potential benefits to others. They only experience the confinement, the fear, and often the pain. Consider the reality of laboratory life for these animals. Often cramped, unnatural environments, social isolation for some, and the recurrent stress of experimental procedures. These procedures can range from injections and force feeding to surgical mutilation and induced diseases, all of which can cause significant and prolonged suffering. I contend that robust ethical consideration should not be a secondary concern, but the primary driver for scientific innovation. A truly advanced society should strive to end its reliance on practices that cause harm to other sentient beings, pushing us decisively towards the development and mandatory implementation of non-animal testing methods. The moral imperative to reduce suffering should be our guide.
Good morning. While Mr. Green has eloquently addressed the profound ethical dimensions, I want to focus on the scientific and practical arguments for moving beyond animal testing. It's crucial to understand that animal models, despite their historical use, have significant limitations. There are fundamental physiological, metabolic, and genetic differences between animal species and humans. These differences mean that results from animal tests often fail to accurately predict human responses to drugs or diseases. This can lead to misleading conclusions, wasted resources, and even harm to human patients when unsafe or ineffective drugs proceed to clinical trials. Fortunately, we are witnessing remarkable advancements in alternative, human-relevant testing methods. For instance, in vitro testing using human cell cultures, engineered tissues, and sophisticated organ-on-a-chip technologies can provide detailed insights into cellular and tissue-level responses. These microfluidic devices can mimic the functions of human organs like lungs, livers, and kidneys, allowing us to study drug effects in a more human-specific context. Computational models are also revolutionizing research. Advanced computer simulations, artificial intelligence-driven predictive toxicology, and QSARs, quantitative structure-activity relationships, can analyze vast data sets to predict the properties and effects of chemical compounds with increasing accuracy. Furthermore, human-based research methodologies are becoming increasingly sophisticated. Microdosing studies in human volunteers allow us to observe how tiny, safe doses of new drugs behave in the human body. Advanced imaging techniques like fMRI and PET scans, along with epidemiological studies that analyze health patterns in human populations, offer invaluable data without any animal use. A compelling argument for these alternative methods is that they are often faster, more cost-effective, and, most importantly, more predictive of human responses than traditional animal testing. Therefore, I advocate for a significant increase in funding, dedicated research focus, and proactive regulatory acceptance to accelerate the development, validation, and widespread adoption of these superior, human-centered alternatives.
Thank you all for your compelling opening statements. They clearly lay out the different facets of this debate. Dr. Hayes, both Mr. Green and Dr. Petrova have raised significant points. Mr. Green strongly questions the ethical basis of animal use, emphasizing sentient beings suffering, while Dr. Petrova points to the growing capabilities and human relevance of alternatives. How do you reconcile the continued use of animal testing in light of these profound ethical concerns and the advancements in scientific alternatives?
That's a critical question, Alex. The ethical concerns are always at the forefront of our minds, and the 3 Rupees framework is our active commitment to addressing them. While alternative methods, as Dr. Petrova described, are indeed promising and are continually being integrated into research, they cannot yet fully replace animal models for all types of investigation. Consider research into complex systemic diseases like Alzheimer's, or understanding the long-term effects of a new drug on multiple organ systems simultaneously, or the intricate workings of the immune response to a novel vaccine. These require studying a whole living organism in its entirety, a complexity that current in vitro or computational models often struggle to capture comprehensively. It's about a, quote, balance of harms, where the potential to alleviate widespread human suffering and save lives is carefully weighed against the welfare of animals used, always under strict regulation and with a commitment to refinement. Many alternatives serve as powerful complementary tools, reducing the number of animals needed, but they are not yet universal replacements.
Dr. Hayes, your balance of harms argument, while common, is problematic from a deontological ethical perspective. This perspective questions whether it is ever truly acceptable to intentionally harm one group, particularly a vulnerable and non-consenting group, for the potential benefit of another. The capacity of animals to suffer is not diminished by our human needs or desires for medical progress. Furthermore, I question the pace at which validated alternatives are being adopted. Is it truly a matter of scientific limitation in all cases? Or are economic factors, regulatory inertia, or simply traditional research paradigms slowing down a more ethically driven transition? If we genuinely prioritize the ethical imperative, perhaps the push for and acceptance of alternatives would be far more accelerated.
Addressing Dr. Hayes' point about the limitations of alternatives for complex systems. While it's true that replicating an entire organism is challenging, we are making significant strides. For example, multi-organ on-a-chip systems are now being developed that can link different human organ mimics to study systemic drug effects, offering more human-relevant data than some animal models. There have been instances where animal models for diseases like Alzheimer's have led to treatments that failed in human trials, precisely because the animal physiology didn't accurately reflect the human condition. Conversely, human cell-based assays have sometimes identified drug candidates that animal models missed. I believe the perceived limitations are often a reflection of underinvestment and a lack of focused, concerted effort to overcome them, rather than an inherent scientific impossibility. A paradigm shift in research methodology, coupled with dedicated funding priorities, could dramatically accelerate the validation and adoption of these more predictive, human-relevant, non-animal techniques.
In closing, I want to reiterate that the medical research community is profoundly dedicated to alleviating human suffering and advancing public health. The ethical discussion around animal testing is valid, and we are not deaf to it. We actively engage in the search for and implementation of alternatives, diligently following the principles of the 3 rupees. However, despite the significant progress in alternative methods, which we welcome and contribute to, animal testing, when conducted under stringent ethical guidelines and robust regulatory oversight, remains a currently indispensable component for certain critical areas of medical research. This is essential to ensure the safety and efficacy of new medicines and treatments before they reach human patients. Our commitment is to both human health and animal welfare, striving for a future where alternatives can fully replace animal use.
I wish to reaffirm the central ethical argument. Sentient animals possess an inherent worth and deserve moral consideration that should preclude their use as mere instruments for research, regardless of potential human benefit. The capacity to suffer is a strong basis for moral standing. I urge for a more compassionate, ethically consistent, and forward-thinking approach to all scientific inquiry. It is imperative that society, policymakers, and the scientific community dramatically increase the priority, resources, and commitment dedicated to the swift development, rigorous validation, and mandatory adoption of non-animal testing methods. True progress lies in innovation that respects all life.
To conclude, I want to emphasize the significant scientific and practical advantages of advanced alternative methods. Their enhanced human relevance, potential for greater speed, and often improved cost-effectiveness. These are not just theoretical benefits. They are increasingly being demonstrated in practice. I am strongly optimistic that a future where medical advancements are achieved without resorting to animal testing is not only possible, but is also scientifically superior. To accelerate this essential transition, we need concrete policy changes, a substantial increase in funding, directed specifically towards non-animal methodologies, and greater international collaboration to share knowledge and validate new approaches. Let us invest in a more predictive, ethical, and human-focused science.
Thank you, Dr. Hayes, Mr. Green, and Dr. Petrova. for your invaluable expert contributions and for such a thoughtful and comprehensive discussion today. It's clear that this issue is multifaceted, highlighting the ongoing tension between perceived medical necessity, profound ethical considerations, and the exciting pace of scientific innovation. We encourage our audience to continue reflecting on the diverse arguments presented here today, and to stay informed on this important and evolving topic. Thank you for joining us.